plastic box and flooding them with water. And we cleaned up the surface and so on. And here you see some small tiny leaves, which are 3 to 5 days old oak leaflets, small oak leaves. And if there is a Phytophora in the soil, they will produce a sporangia, releases zoospores, and those zoospores will swim and get attracted to those leaves. And we will pick up this leaf and place it on a selective media, then they will grow, and here we have the organism. So that's how you isolate. There's no other way, well, there are other ways, but no good way to isolate from soil. Here is the more close-up shot on how we basically collect the soil. And we did a study that had included 2 countries, in this case Austria and Turkey. And we have looked for oak forests. We went to those areas. Some of them were really untouched, but many is really extensively harvested and so on. So anyway, we looked at in Austria and in Turkey, and what we have found was a lot of species which we didn't knew before. And one species, this one here, Phytophora quercina, again, we have described this one. It's a half of the size had this pathogen on it. So looking at the general figures here, this is the rate for Turkey, this is one from Austria, and then there is one that some of my colleagues did in different countries, including Italy, France, UK, and so on. But the main thing was, if you pick up 2 sites, one of them will have Phytophora in it. That's what it says. So they are pretty common, and we didn't knew that before. And besides this one that has been described, we found so many other species too. And some of them are exotic too, that we know. So they were introduced. We did a similar study in the US. Here's our study site, and this is where we are located in Maryland, College Park, in Washington, D.C. So we have surveyed again oak forests. We went to those sites, picked up trees. There are certainly a number of criteria which trees we pick up. But anyway, once we identify the tree, we collect the soil samples, we did isolation, and then eventually get those cultures. And this was a picture for the Mid-Atlantic region in the US. Here are the species again. Some of them we have described as a new species, like this one, SP1, SP2. Some of them are still a wait for description. But one thing that was interesting is this green dot. That was a species which called Phytophora cinnamomai. And this is what we know it is exotic, introduced. And no one knew that actually, that it was so widespread actually. And how did it came to the US and so on? That was the other questions that we asked and looked in the literature and possibly were introduced by the first settlers. So the thing about that particular Phytophora species is it is among the worst alien species list, 100 worst alien species. When you look at the macrofungi, it's not a macrofungi in this case, but under the fungi, you have this chestnut blight or pathogen, you have the Dutch-elm disease, and then here you go, you have the Phytophora cinnamomai. This was the one that we found in the US being so widespread and so common here in the previous picture. This green one, that was Phytophora cinnamomai. It is a very pathogenic organism, but we didn't know that before we did this survey. And this is how it looks like in Australia. It is really moving through soil, and it kills so many plant species, probably 9,000 species in its host list. These are Banksia trees, and this is 1 year movement, 1 year after the other, and when it moves in the soil, it keeps all the susceptible, kills all the susceptible plants. And when you pick up those trees, which is again Banksia, again you see here the images that I showed you at the beginning, all those necrosis, fruit dieback, and so on. So they are, in this case, this pathogen is primarily killing the roots. It's not like Ramorum or the Sutton pathogen. Is there an adaptation to different environments? That was one other thing, because when you look at these red lines, they kind of stop here. What is happening? Why do it stops there? Because you would expect if an organism is so much established, it should move on, and it can probably be present on these areas too. And the best thing really that you can come up with is this climate matching tools. This is the plant hardiness zone map. It determines which plant can grow in which area. If it's too cold, it won't grow. So there are these areas based on this annual minimum winter temperature, you come up with the zones. So Phytophora cinnamomi kind of stopped here in this 40th latitude range. So it was, that data suggests there was this climatic pressure or factor that eliminated this pathogen to move more northern. So there are some, which this data actually is very important, and I will tell you why it is. And we also looked at the cinnamomi in some particular forests in Ohio, and we did a more extensive study, look at so many different trees, and this is also we looked at the moisture index. This red area is dry, and the greener yellowish areas are more wet sites. And we found that the pathogen really liked the areas where it was wet, so which actually we know about in general, but we wanted to prove that. And also we found out that declining trees had a much bigger or greater population of this pathogen. So it was in bigger population, more population of that pathogen, inoculum, infectious part, will kill more roots, and that can contribute to oak decline. That was one of our conclusions. And we also looked at the roots. Trees that had Phytophora cinnamomi in their soil, in their root system, had about 3 times less roots compared to the healthy one, which there was no Phytophora. So we found actually they could kill significant amount of roots. And these were mostly fine roots, which was never documented or never looked at that time. And there are so many other factors, of course, that can play. And it is not only a pathogen's presence. There are so many other factors that feed into the disease epidemiology, which we have also, some of my colleagues did this actually, looked at so many other factors, and each of them has an influence on the Phytophora itself, whether it's going to kill the tree or not. And it is really it can get very complex, as here is the Tokyo metro wave. So you never know where you end up. It's almost the same when we did this study more in detail. Also, you were curious about to look what exists in streams, and here we used the rhododendron leaves, dipped it in the water, and then played it on a selective media. This is how it looks like after 2 weeks, depending on the temperature. And then these things that grow are Phytophora, basically.